All right, thank you for that introduction. Welcome everyone out there in internet land. Thank you for coming late on a Friday night to hear about squid. I'm going to be talking to you about research that I did at Old Dominion University as part of my PhD. I was really interested in looking at how squid evade predators. So I'm going to share with you the data I collected on this topic. So squid belong to a group of animals called the cephalopods. And the cephalopods include not only squid, but also the cuttlefish, the octopus, and the nautilus. These animals have a really robust evolutionary history. They've been around since the Cambrian era, so around 500 million years. So squid themselves are pretty ferocious predators, but turns out everything really loves to eat a squid. So their predators include seals and sea lions. We have marine birds, whales and dolphins eat squid. People enjoy eating squid as well. And in addition to that, fish eat squid and other squid eat squid as well. So everything loves to eat a squid. But they do have this robust evolutionary history. So it's clear that they also have some pretty neat tricks up their sleeve for how to avoid predators in order to survive and reproduce. So for my research, I was really interested in looking at squid from the time that they were little babies to when they were full-grown adults. And there are some pretty interesting differences in squid from the time that they first hatch out of their eggs to when they are full-grown adults. So a baby squid, we call that a paralarvae. And the reason we call it a paralarvae is because it doesn't undergo a full metamorphosis like a caterpillar to a butterfly, but there are enough changes in its body to denote this special terminology. So some of the major differences to begin with are the actual shape of the animal's body. So a paralarval squid has this really short, little round, stubby body. They're very, very small. The species that I looked at was 0.18 centimeters in dorsal mantle length. So that's the length of the body, not including the arms. They also have a relatively large funnel opening for the size of their body. Contrary to that, our adult squid have these long streamlined bodies. The species I was looking at were around 5 to 8 centimeters dorsal mantle length. They can obviously get a whole lot bigger than that. They also have a relatively smaller funnel opening. There's differences in the ecology from when they first hatch out and when they're full grown as well. Our paralarval squid are planktonic, meaning that they are at the mercy of the ocean's currents. They're not particularly strong swimmers. They will, however, have vertical DL migration, meaning they'll move up and down during different times of the day. Our adult squid, however, are really strong swimmers, and they can actually undergo pretty long distance migrations. Another major difference between these two groups is actually the physics that's at play within the water at these different sizes. So our hatchling squid are operating at low to intermediate Reynolds numbers. And if you're not familiar, a Reynolds number is the ratio of inertial to viscous forces in the water. So what is the water actually doing around the animal? How is it behaving? And in our low to intermediate Reynolds numbers, viscous forces are going to be most prominent, meaning the animal's going to feel like it's swimming through honey or molasses when it's this small. Our adult squid, on the other hand, operate at high Reynolds numbers where inertial forces are going to be more prominent. So in this case, our animals are going to be able to take advantage of streamlining and really glide through the fluid. So I wanted to know, at these different life stages, what kind of different anti-predator strategies are they going to be utilizing? So my research was broken down into 3 primary questions. The first being, what sensory systems do squid use to detect a predator? Second being, how do squid behaviorally respond when they detect a predator? And the third being, how does their unique form of locomotion help them to escape a predator? So we'll start by diving into this first question about sensory systems. Squid use multiple sensory modalities in order to navigate their environment. But it's clear just by looking at them that vision is a really important sense. So cephalopod eyes are incredibly well developed. They're very large. They can be, in our giant squid, about the size of a dinner plate and 16 inches in diameter. They are camera eyes, which is incredibly unusual in our vertebrate animals. They also have really well-developed optic lobes in their brain as well. So a lot of energy has been put towards these really advanced eyes. So it's clear vision is an incredibly important sense for our squid. However, they also have another sense called the lateral line analog. Now, if you are familiar with fish at all, you might be familiar with the lateral line system. It runs along the side of the body of the animal laterally, which is why it's called the lateral line. And it's comprised of thousands of tiny little hair cells. And at the ends of each of these hair cells, there's a nerve attached. And when the water moves and pushes these hair cells, it's going to trigger an action potential. And the animal is going to know what direction water around it is moving. And squid have a very, very similar system. It was called the lateral line analog because it is so similar. However, there's one major difference, and that's that this doesn't run laterally along the body of the animal at all. You can see on this baby squid that the arrows are pointing to lines on its head and its arms, and that's where the lateral line analog lies on this animal. So the terminology is a little bit clunky because it was just named to be analogous to fish. Just remember that when we're talking about this system, it's not actually lateral alongside the body at all. But other than that, it's very, very similar. It's comprised of thousands of hair cells, each one that is individually innervated. The literature on the lateral line analogs started coming out in the 1980s, and it's clear that the system is here. However, there's very little information about how it's been used ecologically. We do, though, know a whole lot about how fish use their lateral line system. And what we know is that fish use this lateral line system in order to detect the bow wave of an oncoming predator. And when research was done that removed the lateral line system of larval zebrafish, they were much less likely to successfully avoid an oncoming predator. So knowing that, I was really interested in looking further into the ecological use of the lateral line system and see if that played a role in predator evasion in squid. Now, I was interested in doing this at all life stages of squid. So I had to be a little bit creative. Squid are not an easy animal to work with through and through. They can be really elusive. They also die incredibly easily. So maintaining them is pretty tough. So in order to work through these different life stages, I did have to work with 2 different species. For paralarvae, I used Orthuthis pelei, which I ordered from marine biological labs. They just sent me eggs in a plastic bag, and I hatched them out in the lab. And then for juveniles and adults, I used Loligoncula brevis, which was local to the area where I was collecting this data. So I was able to go out on a boat using a trawl, throw a net over the back of the boat, and actually collect these animals and bring them back to the lab. It's imperfect having these 2 separate species. However, they are very similar, both ecologically and morphologically. So it was the best I could do under the circumstances. And in order to look at the lateral line system specifically, I needed to figure out a way to isolate these different sensory modalities. And this had never been done on squid before. However, it has been done on fish quite a bit. And what they use to do this is antibiotic solutions. So neomycin sulfate was one option. So I had to go and validate that neomycin sulfate would actually ablate the lateral line system of the squid as well. And you can see on the left is a picture of my control group. They're using a 0 micromolar neomycin solution. And the hair cells are abundant, they're nice and smooth and healthy looking. And then on the right is the 500 micromolar neomycin solution, where there was very few hair cells left, and what was left was very damaged. So I was able to say that, yeah, this neomycin sulfate solution does seem to work to ablate these hair cells in squid as well. And then after looking at that, I also had to spend some time behaviorally observing my squid and make sure that this treatment didn't affect them in any way, except for ablation of that lateral line system. And luckily, it didn't. My squid still survived for weeks after treatment, which is a long time for a squid in captivity. They continued to eat normally. They continued to catch prey and behave as a normal, typical captive squid. So I got very lucky in being able to say that this treatment both ablated the lateral line system and didn't seem to affect the squid in any other way. So with that method in hand, I was able to move forward and do some predator-prey trials. So this is what my experiments looks like roughly. I had a basket where I placed the squid inside, and then I had 2 predators. And for the adults, I used flounder, which were actually really aggressive, fantastic predators. I had an acclimation period, and then I would pull the basket up, allowing the squid and the fish to interact. I recorded escaped and survival, and all the interactions were recorded using high-speed video cameras for analysis. And I did the same thing, only at a much smaller scale for the paralarval fish. So I have that represented by the tiny tank up above there. The predators for the paralarvae were little minnows. I did that for logistics for tank size. Also, it's reflective of the predators that these animals are going to face at this point in their life. Now, since I was interested in isolating these sensory modalities, I had 4 different experimental conditions. The first one, they could see and they had their lateral line intact. The second one, they could see, but their lateral line was ablated. The third, they couldn't see, but they had their lateral line. The fourth, they couldn't see, and they did not have their lateral line. And I removed the visual cues by collecting data in completely darkened conditions, using blackout curtains, turning out lights, and then using infrared cameras in order to actually visualize what the animals were doing. So here are my first set of results. We are looking at the mean proportion of escape responses in the 4 different groups. So along the x-axis, we have the light non-ablated group, the light ablated group, dark non-ablated group, and dark ablated group. And then the white bars represent my paralarvae, the gray bars represent juveniles and adults. So starting with the juveniles and adults, you can see that there was a 100% escape jet response in all of the conditions. So regardless of whether they could see, regardless of whether they had their lateral line intact, they always performed an escape jet when confronted with a predator. So that was really interesting. But if we look at the paralarvae, we can see that there's a pretty big difference when we start to remove that lateral line system. So our our light ablated group, when they could still see, but they didn't have their lateral line, is lower than our light non-ablated. And then our dark ablated group is significantly lower than that. So what we wanted to do from here was zoom in on this escape response and see what it is that the animals were actually doing, aside from just were they escaping or were they not escaping. This shows us, that was a reminder for me to say lateral line system seems to be important. So starting with the paralarvae, we analyzed the high-speed video in order to look at the mean velocity of their jets, the peak velocity of their jets, the peak acceleration, and also the time to peak velocity. And there was no significant differences among the 4 conditions within any of these different variables. So it didn't matter whether they could see or whether they could pick up cues with that lateral line system. Their escape jet always had the same mean velocity, the same peak velocity, same peak acceleration, and the same time to peak velocity, which was unexpected considering that we saw those differences in escape response. However, what we think we're seeing here is actually a reflection of the development of their nervous system at this stage in life. So when a squid performs an escape jet, there are 2 different parts of their nervous system at play. There's a giant axon system and a non-giant axon system. The giant axon system is responsible for full-on escape jets, and it's a yes or no response. And the non-giant system is responsible for a more nuanced escape jet. And at this stage, their non-giant system is not particularly well developed, but their giant system is. So what we're getting here is a real stereotyped behavior when they're performing these escape jets. They're either going to do it, they're not going to do it, and when they do it, it looks pretty much the same. Now, when we look at our juveniles and adults, we see something a little bit different. And I will say I had a bit of a research snafu here where I ended up being unable to analyze the kinematics of the animals in the darkened conditions. I simply didn't have enough light to actually collect the information that I needed. But just looking within the lighted conditions, we saw a really interesting trend between our non-ablated and ablated groups. So up in the top, we're looking at the mean velocity of the squid. And you can see that the non-ablated group, the one with the lateral line system, had a significantly higher mean velocity. And then to the right, we're looking at peak velocity, where again, our non-ablated group was significantly higher. Down in the left-hand corner, peak acceleration, where the peak acceleration of our non-ablated group was significantly higher. And then to the right, when we look at time to peak velocity, the non-ablated group was significantly lower. So when the lateral line system was intact for our juveniles and adults, they were faster, they accelerated quicker, and it took them less time to reach peak velocity. So even though in that first graph, I showed 100% escape response, regardless of whether they could see and regardless of whether they had their lateral line intact, if you zoom in on that, there's still actually some major differences between what happens when they have that lateral line system and when they don't. And ultimately, how fast you go and how long it takes you to reach that high speed is going to play a role in survival. So when we take a look at mean proportion of survival in our 4 different groups, again, paralarvae is in white, juveniles and adults are in gray. Starting with those juveniles and adults, you can see that there is a downward trend where we have reduced survival by about 20% when we go from our light non-ablated into our light ablated group, drops by about another 20% when we get into our dark non-ablated group, and is significantly less when we get into our dark ablated condition. And looking at our paralarvae, there's not a huge difference between the light non-ablated, light ablated, dark non-ablated groups, just looking at survival. And I think that is mostly reflective of the fact that the fish could outswim the paralarvae regardless of their condition. But then we do also see a major drop when we get into our dark ablated condition. So based on this information, we can see here that both vision and that lateral line system are playing a role in survival for our squid, both at the paralarvae level and at juvenile and adult stages. So from this chapter, we can conclude that lateral line analog aids vision to support successful predator evasion in squid at all life stages, which was a really neat thing to say, considering this was the first work that really looked at the ecological role of that lateral line system. All right, so moving on to our second question. How do squid behaviorally respond when they detect a predator? Well, cephalopods are infamous for their amazing ability to change color. Up on that top panel is an octopus. You can barely see him in that first picture there. Becomes a little bit more visible in the middle, and he's clearly visible in the last panel. Down below is a cuttlefish who's matching really well with some gravel using a modeled pattern, and then is even trying really hard to match within a checkered board pattern as well. And the way that our cephalopods are able to do this is through organs in the skin called chromatophores. And what you're looking at is a bunch of chromatophores that are both expanding and contracting. What makes the color change in cephalopods so unique in the animal kingdom is that it doesn't rely on a hormonal response like in vertebrates, and instead it's completely nervous system controlled. So each of these chromatophores is a sac of pigment with a musculature ring around it. And each one is individually innervated in order to either expand or contract that ring of muscle around the pigment. So because it's individually innervated, it can create thousands and thousands of different patterns, which is how they're able to match so well with all those different backgrounds. Another anti-predator response that our cephalopods have is inking. So ink is comprised of mucus, which gives it a little bit of a viscous consistency, allows it to have some shape, melanin, which gives it color, and then some chemicals, which we are not sure exactly what they're used for, but they have L-dopa and dopamine in their ink, which are neurotransmitters. They're feel-good chemicals. So this is a common anti-predator response that our cephalopods use. You also see some posturing behavior in these animals as well. This is Lolligoncula brevis, the squid I was working with, and the two most common postures that they would do is spread their arms out really wide. Others would toss them up over their head. Both of these postures were clearly trying to make the animal look a little bit bigger and a little bit more intimidating. So for this set of experiments, I used the same kind of setup as I did in the previous chapter there. I still use flounder as my predators, and all of these behaviors were recorded using high-speed video. So when I looked at this throughout ontogeny, I saw some pretty big differences between the paralarvae and the adults. So our paralarvae only actually performed escape jets in 35% of attacks, which is really, really low if we're looking at an animal trying to avoid a predator. I only recorded one inking event, again, not many, and I didn't see any posturing in the animals at this stage either. So what were they doing? They were doing a bunch of funny little behaviors like this. And it turns out that they rely heavily on the suite of stereotype behaviors This is totally unique to the paralarval life stage. You'll never see a juvenile or adult squid doing things like this. But they would just twirl around in different directions with like no sense of orientation whatsoever. So they would twirl around like I just showed you in that direction. They would do little spinnies. They would dart back and forth all over the place. But it turns out that when you are as small as a paralarval squid and you're working in the low Reynolds numbers and swimming is hard, that it's actually a really good strategy to just be really unpredictable. And that a predator is going to have a hard time interpreting what your trajectory is going to be, what your next move is going to be, if you're just completely random. Now, when I look at the juveniles and adults, it's not so random at all. In fact, it seems like they are incredibly conscious about what a predator is doing and how they're going to respond. So the top graph, we're looking at mean predator distance, and then posturing behaviors versus an ink and escape jet behavior. And white is our juveniles, black is our adults, and gray is a combined number there. And what we're seeing is that when the animal is further away from predator, it's going to rely on posturing first. And then as that predator gets closer, that's when you are going to see an ink and an escape jet. And then down below, we're looking at mean predator velocity, postures, and ink and escape jet again. So the squid is going to posture when the predator is moving slower, and then it's going to ink and escape when the predator is moving faster. So overall, they're going to first rely on posturing and wait and see what the predator is going to do before performing an ink and escape jet, which makes sense because inking is energetically expensive. Escape jets are energetically expensive. So if they don't have to perform those behaviors, they're not going to. And when we look at body patterns, we see different choices amongst our paralarvae in our juveniles and adults as well. So our paralarvae, I categorized into a dark, intermediate, and clear body pattern, and they significantly preferred to maintain a clear body pattern. And that makes sense with those really erratic patterns that they perform as well. They're going to be difficult to track. They're going to be difficult to see when they maintain a clear body pattern and they're just zipping around in weird little ways. When we look at the juveniles and adults, they significantly preferred to have a striped banded body pattern. And it's possible that this pattern breaks up their outline in a way that makes them a little less visible to a predator, or at least surely makes them look a lot less like a squid, and therefore may be passed over by a predator. So overall, our paralarvae use these stereotype behaviors and this clear body pattern in order to be elusive and avoid being eaten, whereas our adults and juveniles show some flexibility in these body patterns, and they're going to save that ink and escape jet for dire situations. All right, and moving on to our third component here. How do squids' unique form of locomotion help them to escape a predator? Squid have a unique way of swimming. Squids swim via jet propulsion. And the way that they do this is they have openings around their head, and they are able to pull water into the mantle, which is the major body cavity in the squid. So they expand their mantle using muscles around it, which opens up that opening, allowing water to rush inside. And once water is inside the mantle, they use the muscle to pressurize it, and it closes the spaces around the head where the water came in. And all of that water is then going to shoot out of the funnel. And they can move that funnel in 360 degrees. So they're able to swim head first, tail first, side to side. They can move in any direction that they want to swim. Now, historically speaking, it was thought that this mode of swimming has to be really inefficient, mostly because it requires a refill period. So they're going to pull that water in, pressurize it, and squirt it out of their funnel. And then they need to slow down and do it again. Bring the water in, pressurize it, and squirt it out the funnel. So it was thought that there's no way that this kind of swimming could be incredibly efficient, at least not compared to a fish that is going to be undulating its tail back and forth constantly without any need for a refill period. But what we didn't know at the time was what flow features looked like. So what does the water do when the animal is interacting with it? In particular, what kind of vortex rings does the animal produce? So this GIF you're seeing is a vortex ring. And when a vortex ring is created in water, it basically entrains more water than the animal was actually engaging with. And when it entrains more water, because of Newton's third law of motion, we actually get a bigger thrust benefit. So in order to really calculate the efficiency of squid escape jets, needed to look at those flow features. And I had to use slightly different setups between the paralarvae and the juveniles and adults because of the size differences. But what you're looking at here is my setup for my paralarvae, where I use digital particle image velocimetry, which is often called DPIV. And the way DPIV works is you have water that is seeded with illuminated particles. And you have a laser that fires at these particles. And the special DPIV camera is going to collect images at the same sequence that the laser is firing. And we then have software that tracks the movement of those particles. Used a very similar technique with the juveniles and adults. The difference between these two, aside from the size, DPIV collects images in 2 dimensions. And then we use defocusing digital particle tracking velocimetry, which actually collects in 3 dimensions. So you can see, again, we have a water tunnel that has been seeded with these particles. We have a laser that's going to be firing at a sequence that's timed with this camera to collect the images as well. In addition to collecting these 3D images of the flow, we did use high-speed cameras just to track what the animal was doing as well. So here's what we found for the paralarvae. On the left hand is the velocity field, and the right hand is the vorticity field. And we ended up categorizing the escape jets into 2 different categories. We had escape jet 1, which was these isolated vortex rings, and escape jet 2, which was a long string of vortex rings. But both of these types of jets we found were incredibly efficient. So escape jet 1 had an efficiency around 95%. And escape jet 2 had an efficiency around 94%. So certainly not the inefficient jet that we thought we were going to see. When we look at our juveniles and adults, we still saw the same 2 big categories, with escape jet 1 being this one isolated vortex ring, and escape jet 2 being this longer string of vortex rings. Now, with the juveniles and adults, the efficiency was a little bit lower than in the paralarvae, with 89% for escape jet 1 and 83% for escape jet 2. But overall, that's still a pretty sinking efficient escape jet. So these animals are not inefficient swimmers. When we actually look at those flow features and include them in our efficiency calculations, they more than make up for that refill period that's needed in order to swim with jet propulsion. So overall, what we saw here was we have these two different approaches, both of which have really high propulsive efficiency. The paralarvae were a little bit more efficient than the adults, and we think that really ties back to the morphology of the animals at these stages. So the paralarvae have a relatively larger frontal aperture, meaning they can move more water out of their body at a relatively faster speed. They also have a proportionately greater amount of volume of water in their mantle based on that short little round body that they have. And then because of the low Reynolds numbers that they're working in, that water is going to move at a relatively lower speed. And in order to maximize efficiency, you actually want to move that fluid at a lower speed and about the speed that you want to be swimming. So all of these things combined make those paralarvae a little bit more efficient than the adults. So I have a lot of people who contributed to this research, all of my committee members, including, most importantly, my PI, who is Dr. Jan Bartal. This work was funded by NSF, by Society of Integrative and Comparative Biology. I also worked with the folks at TED Ed to create a 5-minute animated version of this talk that you just got tonight. I'd highly recommend checking it out on YouTube and also working with them if you ever get the opportunity. It's a very neat experience. And with that, I am more than happy to take any questions that you have.
Fantastic. Thank you for a great talk. And we do have a lot of questions, so I think I'll just go ahead and jump into them.
Cool.
I'll say, if anybody that has more questions or wants to follow up, please do so. Is always more than welcome. So the first question here, this was actually asked on behalf of somebody else in the chat who was afraid it was a silly question, but I think it's kind of a good one.
Silly questions.
Yeah, exactly. Are baby squids reared in any way by their parents?
No. In fact, most cephalopods, they're what we call semiparous, meaning they die after they lay their eggs.
Oh, okay.
Yeah, so I don't want to say there's so many different species of cephalopod, and so I know that there's gonna be some gotcha species out there.
Yeah, there's always one.
Yeah, but for the most part, there's no parental care, and the parents are actually probably dead by the time they've hatched out.
Interesting. I actually did not realize that.
Yeah.
What kind of distances can these squids detect the predators at with the lateral line analogs?
Yeah, that's a good question. I believe it's about for say you have a 1 meter long fish that's swimming towards the squid, they could actually detect that at about 30 meters away.
Wow.
So it's really sensitive.
That's way farther than I would have thought.
Yeah, I mean, they can detect all kinds of little movements in the water, and that's equivalent to what the fish can do as well.
Okay. Are they? Is there any indication that they can detect like differences? Like, do they know that it's a likely predator versus like maybe like a plant or something like that moving through the water?
Probably
I guess
depends.
it'd be hard to tell, but
yeah, it probably depends on the distance of it, and I think that's kind of like where we see those differences in behavioral responses. Like if they feel a little tickle, they might just like make a posture and wait, versus seeing if something's barreling towards them, and then they're like, "Whoo, get out of here."
Makes a lot of sense. How do you actually remove the lateral line?
So with that antibiotic solution, I can show you that real fast. There it is. Yeah. So weirdly enough, antibiotics dissolve lateral line hair cells, and we only know this because of research that's been done on human hearing. Um,
Oh.
the hair cells that are in our inner ear that allow us to hear
Mm-hmm.
are very structurally similar to a lateral line hair cell, particularly one in fish.
Interesting.
Yeah, so this technique has been used in hearing research in order to wipe out those hair cells and to also look at regeneration.
Okay. That's really
I know.
cool.
I did not make up this technique. I never would have guessed that soaking them in antibiotics would be
Yeah.
what we do.
Do we know why neomycin
No.
does it? We just know it does?
Yeah.
Okay, sure. Why not?
Yeah.
Let's see. Has anybody tried to culture the squid the squid hair cells?
Tried to culture them. Not that I know of.
Okay.
Yeah. There has not been a ton of work done on the lateral line system.
Okay.
Really.
All right. So you talked a little bit about the squid's eyesight, or they have their eyes and they're very sensitive. Do they have but you also did the light versus dark in the predator prey models. Do they have like a low or no light enhanced sight at all? Like sort of like cats can see really well in the dark. Is there any equivalent? Okay.
They don't, and I think that's part of the beauty of having the secondary system for them to really be able to feel what's going on around them. Like it's going to be super useful in murky waters and in dark waters.
Makes a lot of sense. All right, I saw this question in the chat, and I figured that this person had to work at least in a similar area, and they said they did. Do the squids have Maltner cells that are related to their escape response like other fish?
Oh, I'm totally blanking on what those do right now. It's like an hour and a half past my bedtime.
That's
so
fair.
I'm not my sharpest right
That's
now.
fine. Insanity.
What?
Could you follow up with what
Yeah,
those do?
remind me what they do.
Yeah, absolutely. We'll get a follow up on that. Let's see. And I appreciate you staying up late.
Yeah. It's a Friday. I can do it.
Yeah, absolutely.
I'm telling myself I'm not too old for this.
Let's see. So, does this is a good question. Does the lateral line system only help in escaping from predators, or does it also help in the capture of food?
That's a good question. It hasn't been looked at how it could help with the capture of food. Yeah, I'm sure it does. I mean,
it would make sense.
Yeah, absolutely. There's no studies that I know of that have yet quantified that. But I mean, I want to say absolutely yes. I'm just a scientist, so I can't without data, but I'm sure.
You have a strong hypothesis that
Yeah.
Let's see. Okay, so you talked about in the inking, I guess, solution that gets released, that has all these different parts, and it has the neurotransmitters. And I believe you said that we don't know at this point what they actually do. Are there any hypotheses for what the purpose might be?
Yeah, there's a few.
Okay.
One is that it could placate an oncoming predator. Like if they got a mouthful of ink, they might feel happy and good and swim on their way. Another hypothesis, which I really like, I'd be interested in looking in further, is that it could be a form of social communication, where this is a chemical being picked up by surrounding squid as well. They can do all kinds of different ink shapes. Like it can be a big smoke screen, or it can just be little tiny little bloops of ink. And I'd be really interested to know how squid behavior changes when they're exposed to those chemicals.
Yeah, that would actually be really cool.
Mhm.
Okay, so we did get a follow up. So Mothner, I hope I'm saying that right. Mothner cells are motor neurons that respond to sound or touch in some fish that are related to escaping.
Um, I don't know that there's anything directly analogous in the squid.
Okay.
Their lateral line, the squid lateral line, is a little bit simpler than the fish lateral line. It doesn't have like the cupula that our fish do. It's literally just hair cells floating in the water.
Okay.
So I don't think so. I also don't 100% know either.
Okay. That's fair. Is inking more common when the squid is solo or in a group?
That's a good question too. Um, the squid that I studied, they swim in schools.
Mm-hmm.
Um, Different cephalopods have different social environments. Like octopuses tend to be more solitary. Our squid and our cuttlefish are a little more social, but that's also a certain type of squid. Our deep
Sure.
sea ones are not that social either. So
Hmm.
I think it probably really largely depends on the species that you're looking at.
That makes sense. Like you said, they probably have different dynamics,
Yeah.
just based on whether they're grouped or not. Let's see. Okay, so are these responses in the lateral line analogs, are they thrown off by the condition of the water? For instance, like if the squid was in rough waters or experiencing a strong current or something like that, well, does that you may not know, like will it change the behavior, but do you suspect that it would?
I suspect that it would. Exactly how they go about like removing background noise.
Mm-hmm.
Um, I'm not sure, and I don't know that it's been looked at yet. Like the same question sort of applies when they're swimming in a school.
Mm-hmm.
Like how do they know the difference between the flaps of their neighbor's fins versus an oncoming predator? And I imagine that their sense of touch is as sophisticated as ours, where like say we're standing in a strong breeze and a bug flies into our face, we still feel the bug.
True.
Um, but figuring out how to remove background noise, that's a really interesting physiological question that, yeah,
Okay.
I don't know.
Let's see. Oh, yeah. This was an interesting question too. What type of sleep, if any, do squids engage in? Like quiet, the quiet or active sleep. The questioner was curious just how they basically can get rest, knowing that everything might be a predator.
That's a really good question. Um, Gosh, I've like never really thought about that before.
Yeah, I hadn't either, but it makes a lot of sense. You like if they can feel everything around them, like how do you relax?
Yeah, I think it must be with our squid, with many of our animals, sleep doesn't look like it does with us, or even in mammals where we like curl up and go unconscious for 8 hours at a time.
Yeah.
But it's more like partial shutdowns of parts of the body and
Okay.
brain. Because they don't like curl up and go to bed.
Yeah. That would be
Like
adorable, but yeah.
it would be adorable. I think I could see like the octopuses having more of a sleep pattern the way we expect it to, but
Sure.
with our pelagic animals, it probably doesn't look quite as satisfying. It probably doesn't. Yeah.
Yeah. Yeah, like I said, we get some interesting questions here.
Yeah.
That's not something I would have thought about.
Yeah.
Oh man. All right. So that was all the questions we have. I'll give a little bit more time in case anybody has any last ones they want to get in. But I will go ahead and say thank you for a really interesting talk. Like I said, this was really cool. And I learned a lot about squids. That's a lot that I did not know, and a lot that I didn't know I didn't know. So like I said, I appreciate you staying up late and agreeing to come on here. This was a lot
Yeah,
of fun. I hope it was a good time for you as well. And
absolutely.
if I could get you to hang around for just 1 second, we'll talk just real briefly, and then I'll let you get to bed.
Sure.
And so I think that's all the questions we got. So I will say thank you to everybody for coming out. I appreciate you, taking some time on a Friday night to come watch and learn about some really cool science. We will be back next week with Jasmine Benjamin from UAB, and she works in biomedical sciences. So I'm sure it will be another really interesting talk. Sorry, we got a quick question here. Random question: can you say something about piglet squids?
They're adorable.
There you have it. Now I'm gonna have to look up what a piglet squid is. I've never. Maybe I've seen one and didn't know it, but
They are worth a Google search.
absolutely okay. Then I'm definitely gonna do it. All right. So we will be back next week. Again, we have Science Palooza 24-hour stream happening July 2nd and 3rd. The link's kind of been put in the chat by the bot a couple of times. If you're interested in potentially giving a talk, check the times, sign up. We got 20, 40, and 60-minute slots. And anybody doing science or science related stuff is welcome. That includes education and stuff like sociology, psychology, anything basically. If it's data driven, it's probably going to work. So that'd be great to have you. And otherwise, have a great weekend. Stay safe. Get vaccinated if you haven't yet. And we'll see you next week. And thank you everybody for coming out.